Thank you. So thank you, Stephen, for this opportunity to talk about ketones. It's a great pleasure for me. First, I have to start that I'm really not a specialist of ketones, so you probably know more about it than me. And I work mostly on Alzheimer's disease, Parkinson's disease, also the blood-brain barrier, and also this connection between nutrition and the brain. But today, what we'll talk about is the quantification of the transport of ketone bodies across the blood-brain barrier. I've been following the field from some distance for the last 20 years. I always felt strange that we didn't know so much about how ketone bodies would cross the blood-brain barrier. And since in my lab we have some techniques to allow this kind of study, we decided to give it a go and try some experiment with ketone bodies. And I will also look at how this transport could be affected by age and Alzheimer's disease pathology. You know these things better than me, that ketone bodies would provide an alternative fuel for the brain, especially the aging brain, that may be more easy to use than glucose, but that to fully do a ketogenic diet is kind of difficult, especially in the elderly. And so there is a need to develop ketogenic products like medium-chain triglycerides, but these formulations have some challenges, but there are some clinical trials going on that you will hear about today. But how do these ketones enter the brain? So how a compound can enter the brain? So there are a few mechanisms. So it can go through. Okay, it's things.
Can you see announcement?
Yeah, I can do that. Yeah. So you have here, oops, the diffusion. So some compounds can freely diffuse across the blood-brain barrier. Some can, well, they have to avoid the efflux system. You have PGP and other transporters that can put stuff out from the brain to the blood, so they have to avoid that. They can use transporter, like for glucose, for example, we'll use GLUT1. Some amino acids also have some transporter for amino acids. So this could be facilitated transport in the case of glucose. There is also what we call transcytosis with a transferrin receptor. This is a bit more controversial. There's also some transfer with channels like aquaporin. There are also more strange way to get across the BBB, which is called absorptive transcytosis. So these are compounds that are charged positively that can get fixed into the glycocalyx and then get across the blood-brain barrier. And you can also induce BBB disruption to open the blood-brain barrier. This can be done, for example, with hypertonic solution. But in the case of energy substrates like glucose will use the GLUT1 transporter. It's a facilitated transport. We get into endothelial cells. It can be transformed into lactate, but it can also get through GLUT1 in the form of glucose inside the brain. But for ketones, it's generally assumed that they use this MCT transporter. And that's what you see in most of the reviews when we talk about different ways to cross the blood-brain barrier for amino acids, glucose, and lipids. For ketones, they typically show this MCT transporter. The reason is that MCT transporter, MCT1, is found in the endothelial cells. So it has been shown with electron microscopy, immunohistochemistry, and more recently with RNA sequencing. So this is an example of RNA sequence data that you can get from the data bank from electron specials. So you see that most of the expression in the brain is really found in those endothelial cells. So it's true, MCT1 is there. But Stephen showed many times in different publications that there is a strong relationship, a strong linear relationship between the level of ketones that you measure in the blood and the rate of ketone entry into the brain as assessed with PET scan. So this linear relationship is not compatible with the necessity of using a transporter. It's more compatible with transport by diffusion. So, but we wanted to study that in a more, I would say, closed model. that I will show you soon. So what are these ketone bodies? So of course, the acetoacetate, but today I will focus more on the D-beta hydroxybutyrate. So these are found, they can be found in two enantiomer, but this one is less found in the circulation. So the DBHB is produced by your liver, converted into acetoacetate in target organs. It's also has a longer stability in the blood, and it's found also in a higher abundance. It's a really small molecule, and it's quite polar. So these kind of molecules are always tricky when you look at the chemistry to deduct whether they will cross the BBB or not. So, like I said, it's been postulated that they have to use the MCP1 receptor, but actually, if you look at data in the literature, there's nothing really clear about that. So what we did is that we used a radiolabeled form of this D-beta hydroxybutyrate, so with a tritium. And this can be bound actually by to the American radiolabel chemicals. So now I will explain you the technique that we're using and why we think we can study this type of compound with this technique. So basically, we inject the tritiated BHB into the carotid. Sure, it's better here like that. So into the carotid, we do some ligature. And the thing is that 100% of what is perfused into the carotid goes into the brain. So it's really 100%. So it's not like if you do an IV injection, only a small percentage would go directly into the brain. All the circulation goes to the liver and everything. And in this case, 100% goes to the blood-brain barrier and expose the blood-brain barrier to eventually cross the blood-brain barrier. So with this method, so you see here, the little diagram that my student made. So you see the BHB that gets, in this case, inside the blood-brain barrier. But we also co-perfuse with sucrose. We co-perfuse, this is a vascular marker. So it's a molecule that will stay into the cerebral vasculature that will not cross the BB. So we can use it as a comparator. So we do relatively really simple mathematics. So we do the concentration into the brain divided by the concentration into the perfusate. This gives you an apparent volume of distribution. So it's a standard pharmacokinetic term. We subtract the vascular volume that we get with sucrose, which is always about 10 or 15 microliter per gram per second. And then we divide, well, just per gram, and then we divide by the time segment. And then we get the coefficient of transport. So it's really a rate. It's a rate of transport. It's a speed. And for the experiment with the BHB, we had to use relatively low concentration at the start because these things cost a lot of money. If you want to do a really high concentration, then you will use the bottle in a single mouse. So we cannot do that. So we have to use a small concentration. But with unlabeled BHB, we can reach much higher concentration. And we, like I said, we use sucrose as a vascular marker, and we can also use an MCP1 antagonist in the experiment. So this is the first kind of data we got. So if on the left, this is the sucrose. So this is only to show that the blood-brain barrier is still okay. So we see that the vascular volume is around, in this case, maybe 13 to 14, but it stays like that even if we add more BHB, even if we add this MCP1 antagonist. So that's a good sign, that's a good start. At least the BBB is okay. So then, first thing to look at is the number here. So we get the coefficient about 1 per microliter per gram per second. So this is relatively, I would say, at the level of something easy to measure, but at the same time, it's relatively slow compared to other compounds. And if we add more BHB, in this case, 200 times more, we see no difference at all in the rate of entry into the brain. So this suggests that there's no competition, that there's no site for which BHB has to compete. So there's no competition. But if we add NCT1 antagonist, we see a small decrease, about 17%. We did this experiment with more concentration of BHB to reach something more closer to what you get with ketogenic treatment, like in this case, 461 micromolar, and still you don't see no difference. So normally you would see a decrease in the entry when there's a saturation, but we don't see that. And this again suggests that there's no saturation. We can also draw a line, and you see it's pretty flat. It stays at 1 microliter per gram. And we can compare this to other numbers that we have generated over the years. So typically, for lipid compounds like fatty acid on the top, DHA, EPA, or diazepam, which is benzodiazepine that cross the blood-brain barrier easily, we get numbers around 40, over 40 microliter per gram per second. With glucose, we get numbers close to 1. But for BHB, it's close to the same number as glucose, but at the same time, while glucose is used to facilitate the transport, if you increase the concentration of glucose, you will see a decrease of the rate of entry. But we don't see that with BHB. So it's kind of puzzling a little bit that it's really efficient, but at the same time, the numbers are close to glucose. Another type of experiment that we typically do with this method is that when we extract the brain, we separate the vascular fraction from the parenchymal fraction. The goal of this is to see whether the signal is all entrapped into the microvessels. So if, for example, you inject a compound and it stays into endothelial cells, you extract the brain, you measure the brain, you say, wow, it crosses the BBB. But actually, it could be only staying in these microvessels. But in this case, like that, on the left, this is the example of an antibody targeting the transferrin receptor, and we see that about 82% stays into the vascular fraction. With diazepam, the fully lipophilic drug, the vast majority is found in the parenchyma, about 78%. But in the case of BHB, it's also like diazepam, but not as much, but the majority is found into the parenchyma. So it suggests that it doesn't just stay entrapped into endothelial cells. So that's for the in situ perfusion, the first part. So then I will show you some data whether ketone bodies, the transport, is it affected by AD neuropathology. So for this, we use a mouse model of Alzheimer's disease. So it's a triple transgenic. It's actually a mouse model of AD neuropathology because it develops plaques and neurofibrillary tangles. So it has both. And they are really nice under a microscope. So, but they do have also a cognitive impairment that you can see that develops with age. And we also use a lot of this model to test different nutritional interventions, like, for example, a high-fat diet to accelerate the pathology. So the first thing we did is to look at the MCT1 level into this microvascular fraction, and we didn't see any difference. There was a lot of variability, but we didn't see any difference for MCT1 in the triple transgenic. But this is only the protein. So when we do the in situ perfusion experiment and we look at the rate of entry of BHB into the brain, again, we see that the transgene doesn't have any effect. So there's no decrease. It's the same with the triple transgenic. However, what we see is a small increase in the rate in the animals that were fed with a high-fat diet. It's not very large, but it was actually quite significant with the multivariate analysis. We also saw a small difference based on the biological sex. The female had a little less rate of entry into the brain, but I'm not sure if it's just an artifact of the fact that the females, mice, don't get much weight with the high-fat diet. So it's maybe just that. But it's still interesting to see that there was a small effect of the high-fat diet. This is only to show that when we do this kind of experiment with glucose, we see a lower rate of entry of glucose into the brain of these triple transient. So that's something we have published before, and other groups have seen too. So that again, there's this difference between BHB and glucose. In this experiment, we compare different animals of different ages. So we see no difference with age. So we start with 3-month-old to 21-month-old. And we did also an experiment with fasting. So we didn't see no statistical difference, but maybe we need to do more animals, maybe because we see a little trend. This is a 6-hour fasting. This is for the mouse. Then I will go for the human data. So we've been collaborating for many years now with David Bennett in Chicago with the RUS-MAP study. I don't know if you know a little bit about that. It's a religious order study in which they follow people that are nuns and priests in the US. So in this study, we have 20 NCI, so controls, 20 NCI, so mild cognitive impairment, with 20 persons with AD, they are age matched. We have the neuropsychical diagnosis, the cognitive scores, and many other information. So we developed, in my lab, Cynthia Tremblay developed a method to start with frozen tissue and extract microvessels from frozen tissue from the human brain. So in this case, we start with the parietal cortex sample. And I won't go into all the details of the method, but basically there's centrifugation, filtration, and we end up with a microvessel depleted fraction, so a parenchymal fraction, and a microvessel enriched fraction, so it's a PDB fraction. And we can look at the proteins and see that, oh, the neurons are in a parenchymal fraction, and the Claudin 5, for example, which is a protein 2 in endothelial cells, is found in this microvessel enriched fraction. And we can also look under the microscope. So we did a few papers on that. I will just show you one example. Recently, we looked at the cerebral insulin receptor in this fraction to probe for an explanation for the brain insulin resistance that we see in Alzheimer's disease. So the insulin receptor is a bit complicated because there's a pro-insulin receptor, there is two different subunits, A and B, and then two alpha and beta, and two subunits, A and B. And what we found actually is a decrease of this insulin receptor alpha B. So this is a subunit alpha, isoform B, but that's the one we found actually in endothelial cells. And that's the one that is decreased in Alzheimer's disease. And it leads to a ratio of A over B that goes high. And this has been associated with insulin resistance in the liver. So that might be a kind of index of insulin receptor, insulin resistance, brain insulin resistance, but found into brain microvessels, not neurons or astrocytes. And this level of insulin receptor alpha B was associated with global cognitive scores. Something now you know very well, there's a reduced brain uptake of glucose in Alzheimer's disease. So I will go quickly into that. It has been shown mostly with the FDG PET, and it's highly sensitive, but moderately specific. We can see it sometimes in MCI and ApoE4 carrier, and has been replicated in various animal models. And it's one of the hypotheses that leads to this idea that there is an energy crisis in Alzheimer's disease. And again, Stephen showed that this doesn't seem to be the case with C11 acetoacetate. So we see this decrease of glucose uptake, but not for ketones. So what we decided to do is to use the method that I just explained to you to look at the microvessel, to look at these transporters. So we look at GLUT1 and MCP1. And first, we have to show that we confirm that these two are highly finding high concentration in microvessels. So we see a lot of GLUT1 in the VA fraction, so the vascular fraction, but almost none in the parenchymal fraction. And the same thing for MCP1. We see it again in the vascular fraction and just a little in the parenchymal fraction. So it's there. And these are pictures also, immunofluorescence showing, these are microvessels, showing that MCP1 and GUT1 can be defined in microvessel of the human brain. And then we can measure them. And what we see is we confirm the general idea that GUT1 is decreased in Alzheimer's disease. So we see lower level of GUT1 in person with a neuropathological diagnosis of Alzheimer's, or in the cases with the clinical diagnosis of Alzheimer's disease for GUT1. And this is really in the microvessels. And when we do the same thing with MCP1, we see no difference. And also, we can do some association. Like I said, we have the global cognitive scores for these participants. And there is a relatively strong association between the level of GUT1 in the microvessel with the global cognitive scores. So the more GUT1, better the cognition. But for MCP1, we don't see any relationship. And for those that like neuropathology, we can go into more detail. So we have the TAS score, the SERAT score, the GRAC score. And it seems to be a little more, the GUT1 seems to be a little more related to A-beta pathology, which can be measured in panel. And also the neuritic plaque with SERAT. But no association with CA. And again, for MCT1, there's no difference. We did also a whole bunch of different correlation analysis. So, I will just show you that GUT1 has many association with different cognitive scores. There's also a strong association between GUT1 and this ratio of insulin receptor alpha A over B, which suggests an association with brain insulin resistance. And there's also an association with neuritic plaques. But you find no such association with MCT1. So if I conclude now, I think I'm probably on time, right? Yeah. So if I conclude on the animal study, we showed that the transport of BHB across the BBB is not saturable, suggesting a slow diffusion. It's kind of strange that the number has to go like that for free diffusion. So that's still something maybe we don't fully understand. And there's also a small effect of MCT1 that's still there. BHB doesn't remain entrapped in endothelial cells, so most of it seems to get an entry into the brain parenchyma. In terms of what can modulate BHB transport, most of the things we have measured have no effect. So AD pathology was not a problem, old age either. Maybe the high fat diet increased a little bit the transport across the BBB. There was a strange effect of biological sex. I'm not super sure, like I said, that it's just a consequence of the fact that the female don't gain weight on the high fat diet, something that has been shown by many. There was no obvious impact of the fasting, and the MCT1 levels were similar across the groups. And for the human data, we see that GUT1 and MCT1 are actually enriched in extract of human brain vessels, so that part is okay. And that GUT1 is decreased or is lower in Alzheimer's disease than controls and associated with neuritic plaque and scar and insulin resistance, but that's not the case with MCT1. And so, oops, okay. So there's a lot of this might explain, it fits well in the scheme in which we say that, oh, there's less uptake of glucose in the brain, in the aging brain with Alzheimer's disease because there's less glucose transporter. But at the same time, for ketones, since, like I said, we don't know exactly what's the role of MCT1, but still we know that the levels are the same. So it's probably not a problem to give ketone bodies to AD patients. So overall, I think that what I just said to you supports the use of ketones as an energy alternative for the aging brain, including Alzheimer's disease patients.